Ocean iron fertilization is a scientific approach where trace concentrations of iron are added to iron-deficient ocean regions to stimulate phytoplankton growth. These blooms naturally draw down carbon dioxide and help show how marine ecosystems respond to iron enrichment and how carbon cycles through the ocean. Across more than a dozen mesoscale iron addition experiments conducted over the past 3 decades, one pattern has been consistent. The ecological changes are temporary. The induced blooms rise, decline, and the system returns to its baseline state within weeks. We see the same behavior in nature. Large, abrupt iron inputs, such as those delivered by volcanic dust during the Kasatochi eruption, produce intense phytoplankton blooms. Yet observations show no lasting ecological effects once the iron pulse subsides. These natural analogs reinforce the understanding that iron-driven changes in the open ocean tend to be short-lived.
If you want, just introduce yourself, your background, and your past in OIF, and then just go into the experiments that you led and co-led, and maybe also dive into your view on the future of OIF and also its misunderstandings or misconceptions.
Okay, all right. Well, my name is Mark Wells. I'm a professor of oceanography at the University of Maine in the School of Marine Sciences. Emeritus now. I've just retired or semi-retired. I'm still doing projects in that, but have stepped away from a lot of the duties at the university. My experience with iron goes back to my undergraduate years when I began working with iron. I had a visionary advisor back then, undergraduate advisor, who thought iron was going to become important, and it turned out was absolutely correct. In my career, I have been on 2 iron enrichment experiments. My first one was IronEx 2, which was the first successful iron enrichment experiment, successful in the sense that we were able to measure a response. I was there as a postdoc, and then later I co-led a iron enrichment experiment in the Western Subarctic Pacific called SEEDS 2 to evaluate what the effect of iron was on a large scale, mesoscale, in that environment. So my background in iron goes back a long ways. I am primarily I sort of spread between biological oceanography and chemical oceanography. I keep a foot in both of those sort of disciplines. So I look at the chemistry of iron and how it interacts with phytoplankton, and how the phytoplankton interact with the iron by changing the availability of iron, by releasing organic ligands and other types of ligands, being molecules that will complex the iron and therefore change its reactivity in seawater in different ways. And it's something we don't really have a very good handle on still, because of let's say avoid the pun, the complexity of the system with all these different types of organic molecules that will bind iron. But we're starting to get a pretty good idea about the overall picture of how iron availability varies in the ocean. And it's not related to the concentration of iron. It's related to what we say, the chemical lability of the iron, how reactive is the iron. So you can have systems which have a fair amount of iron, but phytoplankton can't grow because that iron is tied up in organic molecules that the phytoplankton have difficulty in accessing. They can access it, but it's just very slow for them. So I've been sort of playing in this game for all of my career, of trying to understand this interaction between the plankton and the iron and the plankton. So this brings us to OIF, which is obviously one of the topics that's attracting a lot of interest right now, a lot of attention, both positive and negative, in both situations for good reasons. So the issue obviously is that we have increasing CO2 in the atmosphere, and that's causing changes in our climate. And the question is, what do we do about it? Well, the first thing we have to do, obviously, is cut our emissions. And we haven't been doing a great job on that. We're starting steps now that are going to be very useful, but the current understanding by a vast number of scientists have come together to write a report on climate change. The models are suggesting now that just cutting our emissions is not going to be enough if we want to avoid some of the worst projections of what climate change is going to do. We also now have to extract CO2 out of the atmosphere in order to reduce that greenhouse effect. We don't really have any ways of doing that effectively right now. There are a range of approaches that both on land and in marine, and we call this carbon dioxide recovery methods, or CDR. So what I've been looking at is marine CDR. Can we somehow use the natural system to extract CO2 out of the atmosphere to help reduce the increase in CO2 in the atmosphere? We're not going to actually cause drawdown in the atmosphere at this point, but we just want to slow that increase enough. And ultimately, the answer is going to be to take the CO2 in the atmosphere, liquefy it, and pump it back in the ground where it originally came from in the way of fossil fuels. We have technology where that's feasible now, but there's no way we're even close to the idea of being able to do it on the scales we need or at costs that are anywhere near affordable. That's going to change over the next several decades. Certainly, say by another century from now, one can easily imagine that we will have those technology and that capability. So what we're looking for is, over the next century, are ways that we can try and extract or remove carbon dioxide from the atmosphere in ways that at least will hold it for that amount of time. And so these different marine CDR methods are looking at different ways of doing that. And OIF is one of those ways.
Fantastic. So how much CO2 do we need to remove from the atmosphere? What's your estimate, and maybe how much of that currently is being absorbed by photosynthesis on land, and how much in the ocean, and how much in the ocean specifically is there sort of in-gassing, out-gassing, and where does it come from?
Yeah, that's a great question. So the projections are that we need to remove at this point about 10 gigatons of carbon a year, which is a huge amount, okay? And we have nowhere near that capability at this point. As far as how much is being utilized on land versus in the ocean, that's, you can do those. There are numbers out there that you can do, but they're not particularly meaningful. And the reason why they're not particularly meaningful is because you can't count on those. And if you think back a year ago, 2 years ago in North America, we had horrible forest fires. So one of the main ways that people are projecting to collect, and when you, for example, fly in an airplane and you buy carbon credits and try to offset your carbon that you're releasing, a lot of that is going towards forestation, we call afforestation, where we're making forests that aren't there. So we are taking CO2 out of the atmosphere and making wood, in essence. But it takes a really good firestorm year, and all that carbon goes back into the atmosphere. And that's certainly what's happened in North America over the past couple of years. And the projections are that's going to become more frequent as climate changes. So we can look at the relative amount of carbon going into these different pools, but we can't count on that going forward. So the ocean, though, is responsible for a major part of the CO2 uptake. Okay, CO2 is dissolving into the surface ocean, and that's what's causing another problem, depending on which angle you're looking at, and that's ocean acidification. Because when you put CO2 in water, you basically are making carbonic acid, an acid, which drops the pH. When you drink your favorite pop, Coke, Pepsi, whatever, that has CO2 that's pumped into it, and that drink is acidic as a consequence. And that's part of its feature, actually, is it provides a pleasing taste when you have it. But that's obviously not what we want in the surface ocean. So the ocean is taking up a lot. The ocean is also returning a lot of CO2 to the atmosphere in different places. And to understand that, you have to get into how OIF might work, in that you have phytoplankton that photosynthesize. Photosynthesis, in its basic view, is taking CO2 and water and making organic matter and oxygen. And when that organic matter, some portion of that sinks into the deeper ocean, or let's just say it doesn't sink. When it is degraded, respiration, bacteria break it down, they take the organic matter and oxygen, and they make CO2 and water. So the question is, where does that reaction happen? Well, a lot of it happens in the deeper ocean when these organic particles, the phytoplankton, sink into the deeper ocean. So the deep water is relatively acidic compared to the surface ocean, even today. So what that means is it's storing CO2 in the deep ocean. Well, when that deep water upwells to the surface in various areas around the globe, that CO2 is released to the atmosphere. So we're dealing with a complex system in the ocean where there's portions of the ocean which are taking CO2 out of the atmosphere and portions which are returning CO2 to the atmosphere. And it's the balance of those that's helping drive basically what the current trends are in CO2 in the atmosphere once we start adding the CO2 to it.
And is there currently a net uptake from the ocean? Is it taking more in than out?
Currently, it is taking more in, but there's lots of debate about all of that because the models, look, we have very complex models and excellent scientists that are creating these models, but we don't have good measurements to get all the parameters right. When we're talking about the scales that we're talking about, we'll get some measurements in one site at one time. But if you go back to that site a month later, you may get a different measurement, but we can't get the density of data that we need in order to drive these models more accurately. So you have a lot of disparity among models as a consequence.
In the ocean, what is photosynthesizing? I guess there's like microalgae, there's macroalgae. How many species are there? Do we know how much carbon is it per species, and how long do they store it? What other roles do they have in the ocean ecosystem? And ultimately, what is most promising for OIF? What are we targeting?
Okay, so that's a huge question. As far as the different types, there are, as you say, there's the microalgae, which we call the phytoplankton. They photosynthesize, but they are plankton. In other words, they move with the water. They can change their depth. They can swim in small distances, but they're really not going laterally. They can't migrate, let's put it that way. They just sort of move with the water. Then you have macroalgae, and a large portion of those are generally attached to things, so attached to the bottom. I come from the temperate water, so I think of kelp, for example, which are massive macroalgal species that have holdfast that stick to the bottom. There are some macroalgae which are pelagic that don't attach, things like sargassum. And if people have been reading about some of the problems they're having in the Caribbean area about sargassum weed that is moving on shore. So there are macroalgae and microalgae. As far as the number of species, there are far more species of microalgae than there are macroalgae. There are literally tens of thousands of species of microplankton. And we categorize them in different ways, but one of the easiest is on their size. So we think about the smallest ones, and these are what we call the picoplankton, and they are generally smaller than 2 microns in diameter. And if you want to think of a bacterium as generally on the order of half a micron to a micron, you're dealing with bacterial size organisms. And they're very numerous, particularly in the open ocean. And then you move up into, we call the nanoplankton and the macroplankton, which are microplankton, I should say, and they are generally on the size of, say, 2 microns up to 20, and then from 20 up to, say, 100 microns or so. So you got a range of 2 orders of magnitude in size, roughly, in the plankton. Now, as far as how much carbon do they each take up, it's a function of their size. So you have some species when they're larger, of course, each organism is going to have more carbon. However, the smaller ones, you tend to be more numerous than the larger ones. So when you look at the group of them, the amount of carbon in each of those may not be that much different, just because the numbers of the small ones are much higher. Now, as far as if we want to think of it from the carbon processing in the sense, or carbon cycling, the small ones, basically, you can think of as small things are going to sink very, very, very slowly. And so when they die, the bacteria break them up. The bacteria respire that carbon from the organic matter. They take oxygen, the organic matter, and they make CO2 and water from it. That happens mainly in the surface ocean. And all of the nutrients that they use, when we say nutrients, people normally think about, we call them macronutrients, nitrate, phosphate, and perhaps silicate. Those are recycled in the surface ocean. So you have a recycling environment, we call it. But if you've got large cells, such as diatoms, as pictured behind you there, those organisms are one larger, so they tend to sink faster, but they also make these silica houses, these frustules, which you can see in your diagram there, that little edge there that has some sort of texture to the side. Well, that's basically you're looking through glass, amorphous glass, and that adds what we call ballast, negative ballast to be accurate, which means that it helps it sink faster. So in other words, instead of having that dying organism being broken down by bacteria in the surface water, more of it is being broken down in the deep water. Now, when it goes down below what we call the mixed layer depth, where there's a sharp change in density, we call a pycnocline, or when that cell sinks below that and is recycled down there, that carbon is, in essence, is removed from the surface ocean for some period of time. Because in the surface ocean, the plankton have taken up CO2 to make organic matter, there now is a CO2 deficit in the surface ocean, and so it tends to suck a little bit of CO2 out of the atmosphere. So if you take sinking cells and respire them at depth, in essence, what you're doing ultimately is taking CO2 from the atmosphere. And when this process keeps going, you're basically transferring CO2 from the atmosphere into the surface ocean, where large cells are taking it up and then sinking and releasing it in the deep ocean when they're broken down.
In the past OIF experiments, maybe you could just dive into them chronologically. What was the objective? What basically hypothesis did you have before? What were you able to confirm? What couldn't you confirm? What went well, what didn't, and also what were you hoping to achieve with them in the past? And if you reflect to, if you just compare, I think there was roughly 25 years ago. So how did you think the future of OIF would develop back then, and then what was sort of the outcome, and what do you think now? And
okay, good questions. So going back 25, 30 years ago, more than that, 40, 50, 60 years ago, it was recognized that there are some regions of the surface ocean which have lots of chlorophyll, but also lots of macronutrients left. Okay, so in a coastal environment, when you have spring, springtime comes, you go from winter mixes the water, brings nutrients up to the surface, but there's not enough light for the phytoplankton to grow because they're being mixed down deep. First of all, the light is lower in the wintertime, and I'm talking about temperate regions. The light is lower in winter, there's less light, and the cells are being mixed deeper where there's no light, and so they can't grow. Now in springtime, the sun gets a little higher, the water's warm a bit, the mixing depth becomes a little shallower, and you reach a point now where the phytoplankton have enough light to grow, and there's all these nutrients. And so you have this massive bloom in the springtime where the phytoplankton just grow very rapidly. And the nutrients go down to very, very low levels. And then they stay at low levels all summer, okay? Because basically the large cells have taken up those nutrients and they've sank into the deep ocean, taking a lot of those nutrients with them and the carbon, okay? And all that gets reset in the wintertime with the mixing. Right? So you have this seasonal cycle. And that happens in major areas of the open ocean as well, where you have this springtime bloom, and then as you move into summer, the chlorophyll is still there, but there's not anywhere near as much. Okay, but there are these three large regions of the ocean where that doesn't happen, where you get a small bloom, a little bloom will come up in springtime, but then it stops, and you're left with a lot of the macronutrients, nitrate, phosphate, silicate. So it's a very different system. And those areas are the subarctic Pacific, the equatorial Pacific, and the Southern Ocean. Though all of those three areas are what we call high nitrate, because the nitrate concentrations are high enough for phytoplankton to grow and bloom, but low chlorophyll. They still have chlorophyll. They still have actually a fair bit of chlorophyll, but it's much lower than we'd expect. And so we call these the HNLC regions. Okay. And the question was, why? Why don't the phytoplankton grow and take up all those nutrients in these three regions, just like everywhere else? And so there was a number of different hypotheses. There was that in some areas, grazing was keeping the phytoplankton down. So in other words, like having your lawn where you've got nutrients and the lawn is growing, but you keep mowing it off. And so you never get huge grass height. You just keep it small, right? So that was one of the hypotheses. Another was that in, say, the Southern Ocean, there wasn't enough light. So the mixing depth is still pretty deep in the Southern Ocean in the summertime, and they just weren't getting enough light to be able to take up all the nutrients. And then there was the hypothesis of iron. And that hypothesis goes back to 1933. A scientist by the name of Gran. He proposed that maybe there wasn't enough iron because iron is one of those essential micronutrients. You have nitrate, phosphate, silicate. We call those the macronutrients, but phytoplankton need a lot of other nutrients just like we do. We need a lot of trace elements. Of all the trace elements, we need the most is iron. Iron forms hemoglobin, or is the core for hemoglobin in us, and a whole bunch of enzymes that are possible for me. Well, it's the same for phytoplankton. They don't have hemoglobin, but they have enzymes. Many of them are similar to ones we have that require iron. And so they need a lot of iron. They also need things like cobalt, zinc, cadmium, copper. They need these in small quantities, okay? So these are what we call the micronutrients. Well, Gran's hypothesis was that there just wasn't enough iron in these environments. So people went out and would test this hypothesis where they'd collect water, they'd put it in a bottle, and they collect water in a different bottle, and they'd add iron to one, and they'd look to see whether the phytoplankton grow differently. And they didn't. And they didn't for decades. Okay. And it was in 1988 when a fellow at Moss Landing Marine Labs, John Martin, brought in basically semiconductor industry techniques to make clean rooms which would prevent contamination. And he applied those to study the ocean. And what he showed was if you use clean techniques, you clean your bottles in particular ways, you use a very clean way of collecting the seawater, and then you work with those bottles when you're doing your additions and that, you work with them under a clean environment, suddenly he showed, the phytoplankton grew in these HNLC regions when you gave them iron. But there was a lot of people that didn't believe it because it's a bottle experiment. And he got very angry. He said, fine, I will go out and I'll do it in the ocean. And that's what he did, was the IronEx experiments. Unfortunately, he didn't survive long enough to see the outcome of those experiments, but it showed that when you add iron in these environments, that's what's limiting. So the initial, and then the subsequent experiments in different areas, there's been a total of about 13, were to go around these different regions and see whether the same thing held in all of these regions. Because it's not sufficient to say, okay, we ran an experiment here, so we can apply this to the entire globe. That's not reasonable. So you have to go out and try in these different environments. So all of those previous mesoscale iron enrichment experiments, in other words, where iron was distributed in a patch and it was followed for days to weeks. All of those experiments were to evaluate how much was iron limiting and how much iron needed to be added in order to see the system transform, the ecological system transform. None of those experiments were designed to evaluate whether you could use the iron additions to give you carbon removal. They weren't designed to do that. And in fact, they couldn't do that because the patches were simply too small. Okay. And so you can't even go back and look at those data to understand whether iron addition or how well iron addition might cause or might give you marine CDR. And the big reason why is because, well, two. One is that the patch that they did, sure, we did 8 kilometer by 8 kilometer size patches. This is a big patch of water, but that surface water mixes with the water around it. So you're diluting the iron and you're diluting the plankton. in the patch over time, which is, you can't, if you want to get an idea about how the removal is going to go on a large scale, that you're not going to be able to do that, because on a large scale, if you're in 1,000 kilometers by 1,000 kilometers, you're not getting that mixing. You're not getting that dilution effect. The second problem is that the surface patch in that surface water, it moves relative to the deep water. So with IronEx 2, the experiment that I was on, the patch moved 1,800 kilometers over 2.5 weeks. Okay, 1,800 kilometers with an 8 kilometer by 8 kilometer patch. So where are you going to be in order to measure the carbon export? Because it takes time. The phytoplankton grow up, and when they use up the nutrients, that's when they get unhappy, they get sticky, they form aggregates, and they sink. So you got to have some sort of a sampler that you're running around following this patch in order to try and capture the removal. Well, we don't have tools that can do that, or certainly didn't have it back then.
Did you use multiple boats back then? One
No.
boat in patch, out patch to form like a baseline. Did you verify it by like satellite with the chlorophyll levels to identify the patch, or what did it look like?
Okay, so the first of these experiments went back in the 90s, okay? So we had limited satellite information at that point, but we were able to capture the increase in the chlorophyll in the patch with a satellite. And that, you're absolutely right, it was one ship operations for the majority of these. You basically would sample around in the patch, and then you'd run outside the patch and you'd get a comparator to see how much difference was there. So we were looking then at the ecological impacts of the iron. How does you get a difference in the growth of the phytoplankton? How does that transition into the zooplankton, which feed on the zooplankton? And just how does the system work as a function of increasing iron input?
How many people were involved? How long were you fertilizing? How long were you measuring? How long was the planning for the experiment?
Yeah. Okay. So the planning is years, because you have to write a proposal first, and then you may have to resubmit the proposal. And then once you get funded, then there's months and months of preparation and planning. So it takes a long time to get one of these experiments going. As far as the number of people on it, most of these are one vessel. So even the large research vessels, you're taking on the order, say 30, 35 scientists on board, that are there. There are other scientists that are participating, but they are doing it by water samples that are being collected, stored, and brought back for them. But as far as people on site, you're limited to basically about 30, maximum 35 people. There are some of those experiments, like the Seeds 2, for example, there were 2 vessels, so you'd have roughly twice as many. And in that case, one vessel was used to fertilize the patch and do a lot of measurements. And a second one was used, our vessel was used to go in and do some of what we call process studies to try and understand more of the ecological impact of the iron. As far as the iron addition, how long, that varied among the experiments. So in many experiments, the iron was added twice. But I don't know, there were some that added iron 3 and 4 and 5 times over that time course, over the time course that they were there. But not many. Most of them were single or double additions, okay? Now, as far as how long was it monitored? Generally, it was monitored, depending on the experiment, was monitored somewhere between 2 weeks up to a month or 4 weeks, or sometimes a little bit longer if you had 2 vessels. You were able to do that. You have run into limitations in many of these areas about just how many weeks can you stay out with that number of people. And so you're looking at relatively short-term experiments, at least short term in comparison to what OIF, if it were ever to go forward, would be.
And where did the money come from back then? Because I understand that today the situation is much harder to find funding for like an OIF experiment. And where did it come from back then, and how much was it? Do you roughly know?
Well, yeah, I mean, we are talking millions of dollars in order to run any one of these experiments. Millions of dollars in just funding the scientists to do the work, and millions of dollars to fund the vessel to actually go there and help the work be conducted. Those experiments were funded in every case by government agencies, government science agencies, okay, because these were scientific questions that were being asked. It was being asked to see what's the ecological impact of iron. How does it transform this environment? What is it doing to carbon cycling, not carbon removal? One can imply carbon removal, but we couldn't measure that, as I said. And so that's where the funding came. So your question about where's the funding come now? Well, you see, the funding now to study OIF now is a totally different area. It's not a basic science area. It's an applied science area. And most of the government agencies don't fund applied science anywhere near the same level as they do basic research. And so for me in the US, for example, I would be going to the National Science Foundation for funding. They aren't interested in funding a big iron enrichment experiment to look at carbon removal, in essence, to use it as a tool for mCDR. That's more of a type of funding you'd be going to different locations. So where do you go for funding now? And that depends upon, so if I am part of this group called EXOIS. I'm on the steering committee of this group, EXOIS. And EXOIS is trying to promote the idea that we need to test this idea about ocean iron fertilization. Now, EXOIS could run these experiments next year by going to industry and saying, we need money to do this. Industry will pour in money, okay, to do this. Venture capital, no problem. The problem is that has a potential implication that, well, the outcome is not trustworthy. Because we've seen this before with funding for tobacco research or funding for any of these big things. When they're funded by companies that have a financial gain by depending on the answer going one way, then you're concerned that the results are reliable or the findings are reliable. And so the idea instead is to try and go to philanthropic organizations that are concerned about climate change and want to fund something to do that. And so that's where we're going now, looking for funding, is with more of the philanthropic.
So you need like a billionaire family, or someone with a big heart who wants to make an impact. I'm not sure if you know Dan Whaley. I think he's also in XOIS.
Yep.
And
Yep.
He actually got a very prominent figure to give him money back then. Maybe it was 50,000 or 100,000. Was Elon Musk, world's richest man.
Yep.
So
Yep.
if someone like Elon Musk would step forward today, it would
We can do it.
possibly fund it.
As long as from our perspective, as long as it was clear there was no financial benefit to the individual, depending on the outcome. That's the thing. We want to make sure that there is hands off. So get the money, but don't be telling us what answer we need to get, or don't make it seem like they could be. Okay, and clear. So, for example, the experiments that are being proposed with EXOIS, we probably need something on the order of $50 to $100 million. So that's a big thing. Now, there are people out there that can easily write that check, but we have not located them yet. Let's put it that way.
So what would be the risk of financing it by industry? So basically selling the story of this equity story. Let's say you want to raise a mix of venture capital. So you go to the people of Frontier, which is like an industry organization that helps to facilitate carbon credit sales for Shopify, Microsoft, like a McKinsey, like a consortium of big companies. Didn't really grow, but typically they are able to sign contracts for like 20, 30, 40, 50, I think 60 million was the largest. And the way these contracts work is you only get the money once you deliver. So you don't really, it's not pre-funded. It's more like if you hit certain milestones and everyone's happy, only then we will facilitate or get the money from sort of the people that we advise or the companies that we advise and channel it to you. So typically companies that get a Frontier contract still need to pre-fund the operations. So they need to take someone who wants to bet on your capability to deliver those carbon credits to, let's say, Microsoft. So what they do is they go to a venture capital investor. and they tell the venture capital investor, okay, you can give us 20 million, which we need in order to pre-fund the carbon removal, do the operations, and then we will profitably sell it to Microsoft. And then the investor is basically betting on the appreciation of the equity value, that after Microsoft there's going to be more clients, and basically try to assess if you're like a team that can deliver the service. From what I have observed, most of these methodologies that work are onshore methodologies, and that is also most of the money. So it's a guess, maybe roughly 2, 3, 4, 5 billion have been invested so far into onshore carbon credit methodologies. Most of it, I think, in afforestation, reforestation, these types of activities, and then some that are like these venture capital funded novel methods, like, for example, direct air capture, like sucking out the carbon from the air and storing it underground. And from what I understand, these methods have high certainty in the quantity of carbon that you remove and also in the permanence, but they have low quantity. So they have costs that are quite high. So I think direct air capture sells for like $800 per ton. And to produce, they actually need like $1,600, $2,000 per ton. So they have negative unit economics. So they are literally non-profits, from what I understand. Though they are technically for-profit companies, no one makes money. So if you compare this with like NVIDIA building GPUs in order to create AI, like NVIDIA is printing money, they have really good gross margins, while these type of companies have like really bad gross margins. So it's a horrible business category so far. And the idea why venture capital likes these businesses is because they believe this macro thesis of climate change is only going to get worse. It's a very safe bet. So there will be a demand to remove these 10 billion tons per year. So in OIF, from what I understand, it's the inverse. So you do have potentially very high quantity and very low cost, possibly even positive unit economics, but you have very high uncertainty on the quantity of carbon, because that's basically a range. And maybe also something we can dive in. From what I understand, there's sort of a primary and a secondary carbon drawdown, and both have like different permanencies. So because it's different algae capturing carbon and storing it through different pathways, some through fish, for different time scales, and you basically have this situation where relative in marine CDR relative to anything onshore, it's maybe a fraction of the funding. So it's maybe roughly 100 million, maybe 200 million. So that might be like best case 5% of what the onshore methodologies have received so far. To me, obviously, my motivation is to change this and to motivate more people to participate, because I believe the ocean is the largest possible storage for carbon. I don't see that much space on land, because humans like to grow, we like to procreate, and we need more resources, and it's just going to be more expensive, and possibly the ocean is a better carbon sink than land. And so what I really wonder is so if you think about risks, right? So what's the bigger risk? Becoming like a tobacco company and selling like snake oil that doesn't really help you, and basically lie about tobacco creating cancer and all of these things, which possibly might happen. I mean, I'm not sure how addictive OIF will be overall to the world, but being sort of like a bad actor. Or the other risk is basically that OIF just never happens at all, because you basically say our moral risk is too high, and we'd rather then maybe don't tap this more liquid pool of funding. And just to be transparent for our users, there's companies, for example, that are doing ocean alkalinity enhancement. They recently signed this, I think, $30 million frontier contract They have like a little bit of attraction. I think they have an isometric methodology. So there's like even some like carbon credit registry that says, if you hit these parameters, we will help you sell this to our clients. So there's a little bit of traction in the ocean, though it's minor. So it's not much. Most of it is still on land. On land, there's also this big irony that companies like that do direct air capture, like Climeworks, are actually carbon negative or carbon positive, however you want to frame it. Essentially, they emit more carbon trying to suck down carbon than they. So they're like, it's so costly to set up these sites. The unit economics are so bad. Even the power supply is so bad that essentially it's not really a net negative for the atmosphere. So it doesn't really work. And it actually seems to be the case with a lot of methodologies. They're very like they it's all very frontier. It's all very early stage, and it seems like it's this sort of industry where there already happened a lot of failure, and there will be even more failure in the future. And obviously there's a risk to be associated with failure, or be associated with like over promising and under delivering. And so I basically just wonder, like, why do you or XOS? I mean, you're on the steering committee, so I'm not sure you're like the spokesperson for them, but why do you think the risk of overpromising, or maybe there's a way to not overpromise but still tap this liquidity pool, is greater than the risk of maybe not getting the money at all to be involved in this?
Right. Okay. So you've highlighted a fundamental difference between land-based CDR and ocean-based CDR, or marine-based CDR. On land-based CDR, the techniques are either what most people would consider positive, in the sense you talk about afforestation. We're planting forest. Well, that's good for the environment if we plant a lot of forests. So there's very little risk of things going bad in their eyes. Okay, that may not be the case, but in their eyes, there's not much risk. When you talk about direct air capture, yes, I mean, that's ultimately, as I feel, that's the ultimate answer we're going to have, but it's going to take us decades before we can get to the scale and the economics to make this feasible. The difference is when you go to the ocean is people are concerned about the impact on the ocean system, on the ocean ecology, on the cultural components, the economic components, about how the ocean is being used. And so there's a lot more wariness. Combine that with the fact, as you point out, there's a lot more uncertainty about just how well might this OIF work. So this was proposed about a dozen years ago. We should be looking at OIF as a way for CDR capture. I was against the idea, because I felt that the real answer, and still the real answer, is cutting back our emissions. We have to do that. And so at this point, it's premature to be looking at carbon dioxide recovery, because we've got time to cut our emissions and achieve the goals that we want. Well, that was a dozen years ago, and we all know how well that's gone over the past dozen years, and it's not likely to change over the next dozen years. So that's what's caused me to change my view and say, not that OIF is a good idea, but that we need to look at it. Manipulating the ocean system to achieve a particular goal is a bad idea. But that's not the important question. The important question is it the worst idea? Because we know that the changes that are coming with ocean systems are going to be massive if we do nothing, if we don't capture carbon. Now, as you pointed out, I think, is that land capture. So first off, there's no single method that's going to be able to achieve the capture rates that we need to capture. It's going to have to be a consortium of approaches, okay? No single one is going to accomplish everything we need. Of the ones that are out there, the one which has the greatest potential for the magnitude of drawdown is OIF. Ocean alkalinity enhancement would have a potential that would be near it if one could apply it across a large enough region, and do it with a more carbon neutral approach. And that's something which is a big problem, because for ocean alkalinity enhancement, you're going to have to mine it. So you have the impacts on land, you have to transport it, and then you have to distribute it in a way where it interacts with the atmosphere. Now, I am not against OAE by any means. I think that all the research has to go forward on it because I think it has a place in mCDR, okay? But it's not going to be the answer that can help with this. We're going to need other methods. And ocean iron fertilization has a greater potential at a much lower cost to implement this. But is it a good idea? Is it an idea that we can live with? And right now, we can't say yes or no. People will point to the previous experiments and say that, well, carbon capture doesn't work. Well, we didn't measure the carbon capture back then. We can't use those results in order to figure out whether carbon capture will work. Our models, you can create the models and put different parameters in, and you can make them either work or not work, but we don't have the actual data in order to best parameterize the models to answer these questions. So what I'm feeling, and what is exuous, is that we need to run experiments that will provide the models the data they need so that we can project what would these impacts be over the long term with some level of certainty. Because right now, we're just waving our hands in the air primarily, okay? Because we have to pull parameters out. We have to guess at a lot of things. So, I don't think that OIF is a good idea, but I think that we need to look at it because the consequences, if we don't get enough carbon removed out of the atmosphere, in my view, could well be worse than us doing OIF. There's many model projections that at least maybe not quantitatively are going to be accurate, but certainly are in the right trends. And we're going to have decreased ocean productivity moving into the future because of warming of the surface waters. We're going to be losing fisheries because of that. We are going to have transformations in society beyond just the ocean unless we take CO2 out of the atmosphere. And so my personal view is, let's get some answers about whether OIF might work. So that comes back then to the funding question. We don't want to go to venture capital to fund this because venture capital, as you point out, is looking to get a return. They're looking for a specific answer. We're not going to give them a specific answer. We're going to go out and experiment and find out what the findings give us. We're not going to have a venture capital group or an industry group pushing us to shade our interpretations in a way that's more positive for their financial outcome. And that's where the difference is between land-based and ocean-based. Because land-based, the techniques, at least in society's perspective, the techniques are fully acceptable for the most part, right? You're planting forest, you're direct air capture. You don't have big facilities. This hasn't got a big negative environmental impact. Okay. The ocean, on the other hand, there will be some negative impacts in different aspects. There'll be perhaps positive aspects in other components, but the balance of those, we can't tell people what they are yet. We just don't know. So that's why these experiments, I feel, have to be run.
So let's say you meet someone who's very rich, who gives you 50 to 100 million, and you do the next experiment. From what I understand, it is planned to do in the North Pacific. And what are the parameters there that you are most curious about that have not been measured and answered before? Maybe my personal point of view, and I have only studied this topic for maybe 2 or 3 years. What I always found very interesting is the measurement of a marine algae called Chaetosaurus sinking down. So it's like measuring its growth at the surface and then measuring it sinking down a few hundred meters deep so that you could assume that it would sink until the bottom of the ocean. But then at the bottom of the ocean, it might just be eaten up by like an Arctic krill or something. And then the question is what happens to the Antarctic krill, and then it's the permanence is still a question. You can't measure it forever. You can just measure it for a few weeks. You measure it growing, you measure it sinking, but then you're just going to lose track of the carbon, which is harder than if you mineralize carbon in direct air capture, or if you grow a tree, you can look at your carbon every day. You'll lose your carbon. So how do you want to measure the carbon with a 50 or 100 million? And what's the most unanswered question in that specific region?
Okay, so let's go first to the question of the region. So why the subarctic Pacific? Well, that's one of those HNLC zones. Now, the reason why we've selected the subarctic Pacific as the first target area is not because it's going to be the most effective at carbon removal. The most affected is probably the Southern Ocean, but the Southern Ocean is also the most difficult place to try and study and run one of these experiments. And it's going to be a complex undertaking in order to be able to get the measurements that we want. So as a first order, a first test, we thought experiments in the subarctic Pacific would make sense because it is on an ocean basin scale, it is a relatively quiescent region. The surface water does not move that fast relative to the deep, which means it makes it simpler for trying to capture the amount of carbon that is actually sinking. Whereas in the Southern Ocean, the surface water is moving quickly. And so now you have to, and in ways that are not necessarily fully predictable. So it's much harder to measure that export when you are trying to get a good quantitative measure of what's feasible. Okay. So the subarctic Pacific made the most sense. It also, they've got experience there from there's data sets that run back decades from long-term ocean observing stations there. So we know what the background looks like in that area very, very well. There's also been an iron enrichment experiment done there. So we know what the general outcome is. We've also been out, I've been out there running cruises and experiments for decades. And so there's lots of deckboard experiments that we understand more about that system. So there's a lot of information that allows us to help set up the experiments. And logistically, it's close to ports, close to ports in Alaska and in British Columbia and in the US. So it makes it logistically more straightforward to do. So that's the reason why the subarctic Pacific was chosen. Now, what are the important questions? Well, they basically come down to functionally, how effective is the carbon removal? And by carbon removal, it has to go past that deep mixed layer. Okay, it has to go down to a depth where that water will eventually interact with the surface again. Okay, but we want it to be down there for at least 100 years to give us that breathing space, literally, until direct air capture or other methods become much more feasible and effective on the scales that are needed. Okay. So we say that we want to plan for 100 years of removal. So we have to get there in the subarctic Pacific. We have to get particles or cells to export deeper than 500 meters. So we want to know, what is that export across that range? And at what depth are those phytoplankton cells remineralized? Because if they're remineralized shallower than 500 meters, that carbon, that CO2 that's released as a function of the respiration of that material, that conversion of the organic matter to CO2 and water, that CO2 is coming back to the atmosphere in less than 100 years. So we want to know about what that impact is, okay, from the carbon export. Second is, we want to know what is the effect on the amount of oxygen utilization, because one of the big concerns, justifiable concerns with ocean iron fertilization is, well, you're moving more carbon into the deep ocean. Actually, I should break this up. You're moving more cells into the deep ocean. So there's more respiration in the deep ocean, which means you're using up more oxygen in the deep ocean. And deoxygenation is a potential problem. Now, most of that deoxygenation is happening in above 500 meters. So we could make that much worse there, to the point where it has a negative impact on the ecology. And how big an impact, that's the sort of thing that we would be measuring by looking at different biological and different chemical parameters. To be effective, to engineer this well, then you want to actually reduce the amount of deoxygenation above 500 meters, because you want most of that to happen below 500 meters, because you're then got a much more efficient export. well, then you may end up having deoxygenation. Let's go to one end of the possibilities. You make all this bloom and you package it up in a great huge ball that sinks like a stone to the bottom in 4 days. And now you ended up with all this organic matter on the bottom, and you have all this respiration. You have deoxygenation. You're going to kill most of the benthos down there. So you don't want that. So the alternative is, and the ideal would be to see that deoxygenation is spread out across the entire water column, the 5,000 meters of water, in which case the impact at any one depth is relatively small. So we're looking at ways of trying altering that export efficiency to, if you want, smear out that deoxygenation across the depth. So that's going to be one of the important factors. And the other big factor is obviously, what is the ecological impact over the course of the year? So most of these experiments, that we said, have been done for maybe up to 3, 4, 5 weeks. We want to be measuring over a period of 3 or 4 months to say, what is the impact across this whole seasonal time cycle by running this type of experiment, so that we have a better impact or better understanding of that impact in the longer term. So those will be the major questions that we'd be looking at. There's a bunch of others, but those would be the major ones.
So I remember that the current thinking is, or the hope is that, please correct me on the percentage, but that there is, in the open ocean, that there's a lot of oxygen, and that the possible impact on oxygen levels would be something around like 3% negative. So if OIF works, if the parameters are sort of confirmed in this experiment, you wouldn't take out more than 3%. Is that roughly correct?
Well, okay. There are models that show that. We have a model. I'm working with colleagues. We have a model where it's less than that. But it all depends upon how the model is parameterized. That's the issue. And none of these models we can say are parameterized wrong. We have feelings about that maybe that's not as reasonable, but that's a scientific debate. There isn't a right answer at this point. That's the reason why we have to make these measurements. The other factor to keep in mind is that deoxygenation, I mean, you can calculate the amount of carbon that you're going to export, all right? And you can come up with how much oxygen is going to be consumed by respiring that. The question is, where in the water column will that happen? If 90% of it happens in the upper, say, 200 meters, you're going to have a bigger impact there than 3%. Okay. If you spread that out across more of the water column, you're going to have a much smaller impact at any one depth or one location that you're at. And so that's where these models right now can't give us a good measure of what's going on, because they are working on how a natural system responds when we're creating an unnatural bloom. We're creating a bloom that's bigger than normal. And so its behavior from the point of view of export is going to be different. And what we don't know is how different will it be.
So in terms of other ecological consequences, one of the big topics that we've discussed in this podcast is this misconception or misunderstanding about toxic algae. So there are toxic algae blooms sometimes in some coastal areas. And I think Smedachik explained that I think there were a group of senior people that once got sort of intoxicated, and some had like a weak immune system. I think 2 people died from like seafood with the toxic algae blooms, and I think there's seals or dolphins that also suffered under like toxic algae bloom, but it's a relatively small amount of toxins historically, and you published on that topic, and from what I understand, your research was also misunderstood, maybe intentionally or unintentionally.
Yeah. Okay. So, there are. For many years, we recognized that, well, it goes back in time, recognized that there are times when the seafood becomes toxic and kills people. All right. For example, on the coast of British Columbia here, the indigenous peoples go back, of course, centuries, they have in their records what they would do is in the summertime, they would post sentries at the headlands, and they would watch at night. And if the water bioluminesced, they wouldn't eat the shellfish. And it turns out that among the cells that bioluminesce, not all of them, but among them are species that produce potent toxins that will kill you in a short period of time if you consume enough of that toxin. And the shellfish will bioconcentrate these toxins. They are what we call lipid soluble, or they're fat soluble. And so they basically will be eaten by the shellfish is filter feeding the cells, the phytoplankton. They retain the toxin in their tissues and they bioconcentrate it. So if you wanted to drink a liter of seawater with these cells in it, it would not affect you because the concentration in a liter of seawater, even in all these cells, is so low, it would not reach a point where it's a problem. But if you eat a shellfish, which is filtered hundreds of liters, thousands of liters of seawater and collected all of that toxin into that one little parcel, that could kill you, okay? And there have been, but those organisms were primarily recognized as dinoflagellates, primarily a group of phytoplankton that are very different than diatoms. Diatoms are always viewed to be safe until 1988 or 87. I think it was 87. There was a bunch of people that died in Prince Edward Island, Canada. And I shouldn't say a bunch. There was over 100 people were badly sickened. I think 2 people died, and they were eating bivalves, but there was no dinoflagellates. And what they did in a very short period of time was the scientists figured out that actually it was domoic acid was the toxin, which was being produced by a diatom. And domoic acid basically mimics glutamic acid, or is very similar to glutamic acid, and it binds to the brain and it kills brain cells. So it's called amnesic shellfish poisoning that you get. And if it's bad enough, then you die from the degradation of the brain. So for many years after that, and we get blooms of these toxic dinoflagellates, diatoms along the west coast of the U.S., and we've been starting to get them along the east coast of the U.S. But the west coast, we've been having them for decades. And now we recognize we've been having them for decades. Fortunately, people weren't getting sick because not a lot of people were eating shellfish off the coast there. But it's recognized as being a major problem in the near shore, the shelf environment. But at that time, for years, it was thought that the offshore phytoplankton, the offshore Pseudonychia, because there are the species of diatom that produces is called Pseudonychia, the genus. There is a lot of Pseudonychia species in the open ocean, but it was presumed that they didn't make any domoic acid. So we went out on one of these experiments in the subarctic Pacific, and we tested, we added a bit of iron, we got diatoms to grow, and we tested for domoic acid, and we were able to detect some. And so our recognition then was that, well, the offshore species can produce domoic acid. So if ocean, and even back then there was thought that, well, they might be doing iron fertilization, then we should be thinking about this rather than ignoring it. And so the paper that we published back in 2010 was essentially to give this message that, hey, we can measure it. The amount that we measured was so small that there's no way it would ever cause a problem, even if shellfish were there feeding, okay? But the point was that we could measure it. And so that you should be looking to try and at least measure the presence of these cells and the domoic acid if you're going to run these iron enrichment experiments.
So in the end, to conclude, it's probably about the dosage, and most of the dosages that you'd get isn't harmful to a healthy human being.
And in our case, it wasn't. Yes, but that's not to say that it might be worse in a large scale experiment. We should be at least measuring the presence of it.
My assumption is that most of the negative reputation of algae blooms comes from fresh water and coastal areas, because most people have seen an algae bloom in a lake, in a river, around a coast, and it's typically being perceived as negative. And say, this is dirty water, this is we don't treat the water right, and some is from fertilizer. There's also other sources that create them. So that's why most people are sort of scared about algae. I think some, also rightfully. I think there's even a big problems that can occur if you go swimming in a lake with some specific toxic algae. But most humans don't really spend much time in the Southern Ocean. There's not much happening there. So, and from what I know, there's also very little fishing happening there. So it's actually a region of the planet that is like, sparsely populated. So from my understanding, if OIF were to roll out and you ultimately get into the Southern Ocean, this is something you should control and measure, but it's very, very unlikely to be a problem.
Well, okay, that would be nice, and I agree. Well, first off, there is a lot of fishing in the Southern Ocean, but and your comment about algal blooms, I think, is right on for a lot of society. But what most people don't understand is that phytoplankton blooms in general are essential for our well-being, because those blooms are what generate a lot of the energy that moves up into the trophic levels. So it drives a lot of our fisheries, a lot of our other things. So blooms are a critical part, a natural critical part of the system. Where you're talking about lake systems and that's usually areas where humans have put in more nutrients or changed the system in ways that facilitate the bloom of a particular type of organism that can cause us problems. But natural systems they depend upon algal blooms happening. So when from the point of view of harmful algae, I'm not going to say that OIF won't cause a harmful algal bloom. Okay, I'm going to say that the evidence, the single set of experiments that have been run so far, suggests that it won't. But you're never going to want to make any firm conclusions based only on one experiment. It needs to be repeated. It needs to be looked at in a broader scale. And our experiment was done in a bottle. And bottle experiments give us a lot of good ideas of what may be going on in the natural environment, but it's not an exact replication of what's going on in the natural environment. So that's why I will argue that you still need to be looking for the presence of these toxin-generating organisms and the amount of toxin that they happen to produce. Because that's one of the other factors that is interesting about these toxic diatoms is they don't always produce the toxin, or at least many of the species don't always produce the toxin. And so, it really depends upon the circumstances.
Understood. We also ran a lot of bottle experiments when we were researching algae in fresh water in lakes and rivers, and it's very hard to, there's so much contamination, there's so many different factors that you can't really assume that would happen in open water systems the same that they would in the lab.
That's right.
So it's a very replication rate quite low. Or maybe to talk about a few more of the other common misconceptions, I think one big one that people are concerned about is what's called nutrient robbing. So the idea that nutrients in the ocean are limited and that we would exploit nutrients for the benefit of our climate sins. So we would basically, as humans, emit a lot of carbon, and we then would steal those nutrients to sink the carbon in the ocean, and that would ultimately be bad for fish and other people that need those nitrates, phosphates in the ocean. So from my research, there's areas of the ocean that are, from what I understand, around the Southern Ocean, a lot of those nutrients basically are sort of rotating the Antarctic continent. So some of those nutrients aren't really becoming productive right now because they're sort of like circulating, and that's why, in my estimation, the Southern Ocean would be just using an unused resource, a resource that otherwise is only partially being, these nutrients only partially reach the coast of Argentina and then become productive, because then there's some iron source suddenly with like the Patagonia Desert or with the landmass itself. So
Right.
and then maybe there's like a 3% loss or something, or there's some estimates that you'd lose some of those nutrients that otherwise would have been productive, but largely those nutrients are unproductive anyway.
Yes, well, that's the whole thing about the HNLC zones, those high nitrate, low chlorophyll. So there's high nitrate, there's high phosphate, there's high silicate. When we say high, there's still micromolar concentration, so still really low concentrations, but it's high relative to what we would expect there to be under a normal bloom situation, say a coastal environment. So the nutrient robbing is going to be a key issue that has to be understood before any sort of large scale implementation happens. Okay, so what we're talking about now is running experiments to understand how the system would respond, to be able to give us data, to be able to make projections, to give us the information we need to decide whether or not it's a good idea to implement OIF for carbon removal. Okay, so that's what the experiment, that's the stage we're at. Now, with nutrient robbing, as you point out, the concern is that you may end up use up nutrients that later in the season or downstream of your area would then end up with impacts later on. Okay, so that's why I say for these experiments, we want to run over a period of 3 to 4 months, because then we can examine what the impact is downstream or with time in the patch area to understand what that effect of the drawing down those excess nutrients will have. Just realize that for a lot of the ocean, the majority of the ocean, the nutrients are drawn down in springtime and stay low in the springtime. Or after spring, I should say, over across the summertime. So it's not like we're creating a system which is totally different than a natural system. It's just that we're making it go that way in an area that it's not done it before. So what are those impacts? How might it change the ecology and the productivity of the ocean? The Southern Ocean, you're right, you have the circumpolar current, the Antarctic circumpolar current that's spinning around the Antarctic continent. Okay, and if you're staring at Antarctica from the South Pole, it's going in a clockwise direction. But what's important there about the nutrient robbing is that surface water ends up being subducted and going to higher latitudes. It's called the submode water, Antarctic submode water. And that water stays close to the surface as it moves northwards. And so a lot of the nutrients that end up going into the lower latitudes in the surface ocean, and we're talking about the open ocean now, a lot of those nutrients came from Antarctica. So if we start cutting down the amount of nutrients in Antarctica, we may have downstream effects at higher latitudes because of that connection. And so that adds another level that the only way you're going to be able to get at this is by doing modeling. And the only way you're going to be able to do that modeling is by getting more accurate parameterization about how much of the nutrient is going to be actually drawn down, how much is being recycled in the surface. Is that recycled nutrient in the surface is subducting and moving north, and how much of it is going down deep. And it's not there's no simple answer to this. And so that's going to be an important issue. But you have to keep in mind that we are going to have nutrient robbing, even if we don't have OIF. And the reason is because the climate is warming. The atmosphere is warming. And as the atmosphere warms, the surface water warms. And as the surface water warms, the density difference between the surface and the deep ocean becomes greater. That picnic line, that connection between the two, becomes much more of a barrier for nutrients moving from below into the surface ocean. So there will be nutrient robbing without OIF, with climate change progressing. The question then is, will it be worse if we use OIF and slow climate warming? That's the ultimate question that only models are going to be able to tell us, but we don't have the model parameterizations yet that will give us those answers.
So from the total potential of OIF, from what I understand from Victor Smetacek, was maybe initially 2 to 4 billion in the Southern Ocean in terms of carbon. And I think that's an estimation of a simple equation of what's the surface area and what's the density of diatoms that could grow at this surface area. And then maybe afterwards 1 billion per year, like one gigaton per year
1 gigaton.
by
Yeah.
and it's like the high silica region where there would be like a lot of diatoms and some like direct carbon drawdown. And then this would exclude all the other areas of the ocean. But what I also understood from Smettachek that we do have more nutrients in the deep ocean. So like the deep ocean is sort of like nutrient rich, and I think there's all these ideas about like
Yep.
we could upwell those nutrients only to then make those nutrient productive again with algae blooms at the surface. And I always wondered in this upwelling of nutrients idea is if we upwell nutrients, or if we take water from the deep ocean, we also bring the carbon to the surface. And is there actually a net? So what's actually the net effect? Can you actually only basically get the nutrients up without outgassing a lot of carbon, only to then like ingas the carbon again with a diatom bloom?
Yeah, that's great. Great question. And that's really the uncertainty about ocean upwelling systems. The artificial upwelling is that, yeah, you bring nutrients up, but you're also bringing up a lot of that water where the nutrients are there because you've had respiration breaking down organic matter. So you've taken the organic matter and oxygen and you made CO2 and water, and you put out a lot of nutrients, but you put CO2 in that water. And when you bring it to the surface, that is super saturated relative to the surface conditions. So that extra CO2 is going to go back to the atmosphere. And the question is, what's the balance of those? And we don't have an answer, okay? Because it's going to depend a lot upon the nutrients you bring up. How efficient, effective will that export be? So it's the same issue as you're dealing with OIF. We don't know how effective it can be, okay? So there, for example, if we use natural numbers, which is what all the models are doing now, you can end up with a significant portion of that carbon being exported below 500 meters, okay? But there is examples that are being used now in coastal environments where that export is being enhanced, okay? So you're using an added technique to try and enhance export. And that added technique that people have worked on a lot is adding clays. Small amounts of clay into the surface water helps things aggregate and gives you more negative ballast, and so you end up having faster sinking rates. So you can have more effective removal, okay? And so none of these are factored into any of the models. Whereas if you're spreading iron, you might then, after the bloom builds, spread a little bit of clay, and now suddenly it becomes much more efficient at removing things. So that might be the sort of thing that could happen if you did ocean upwelling. Enhanced ocean upwelling, but that's far beyond the issues that we're thinking about right now. One of the big issues about this is that for the magnitude that you'd need to do this, all right, when we're talking gigatons, for ocean upwelling, they have ocean tubes that they will use. Okay, and they've run experiments with these where they put these tubes down and they can be operated by wave action. Basically, they get a natural pumping, so it's low energy to have this all go. Well, that's great. You have thousands of these tubes out in the ocean, and they're all subsurface. You're not going to run into them, okay? Which is part of the problem, because if you decide, wait a second, we're having negative impact here. We're seeing the start of a negative impact. We want to stop this we want to stop this enhanced upwelling. Well, trying to find and remove all these tubes, it's not logistically feasible. Okay, these tubes we're talking about are maybe 100 meters long, and there's thousands of them that you put out into the ocean, and you have to find them now because they just drift. And so turning off the systems starts becoming just as important a target or goal of any mCDR. Being able to stop it is going to be critical. Ocean alkalinity enhancement, you can stop adding the alkaline substances. Okay, ocean iron fertilization, you stop adding the iron. With some of these other techniques, it's not so simple. It's not so easy. Okay, for example, with macroalgae, you could stop producing the macroalgae, but a lot of those the macroalgae, it takes time for it to degrade. So you may be looking at a decade or more before you actually stop the process, which can be perhaps a problem. I'm not saying there's anything against using macroalgae or anything else. It's just something that has to be considered when designing the type of system. Okay, and that's where the enhanced upwelling starts becoming problematic, at least if you want to try to do it at any scale.
Another thing that always comes up is the toxicity of iron itself. So if you add iron to the ocean, how quickly does it mix? Like for what amount of time is there like a high concentration of iron in the ocean? And I don't know, from how many hours or days does it take for it to be so diluted that it's basically lower than the recommended EPA toxicity levels?
Yeah. Okay. So it would be, I hate to say the word impossible because there's always a way somewhere, but it would be virtually impossible for iron to reach levels that would be toxic to any organism in the ocean. Okay. Um, at least the organisms that we're aware of, certainly phytoplankton. All right. And the reason is because iron's not very soluble in seawater. So you add iron and it in a dissolved form, and this is part of the challenges of OIF. You add the iron in a dissolved form, and the concentration goes up to a certain point, and then suddenly now, if it goes higher, you're above solubility, the iron precipitates and then sinks. And so you are capped based on the solubility of the iron. And the solubility of iron is so low that it's not going to be toxic to any organisms we're aware of. So there is no toxicity of iron. If you add too much iron, which is not something you want to do with OIF because you're spending a lot of money to get the iron out there. So you don't want to add too much. You want to add the right amount in order to get the response you want. Okay. But if you were to add way too much iron, you get a lot of precipitation of iron. You have the potential to perhaps strip out some of the other metals that are essential, okay? Because iron's very surface active. So you can have absorption of metals to that particle, metals that are utilized by other phytoplankton, and they would sink out. So you would be stripping some of the other micronutrients out. But in order for that to happen, you're going to have to add a huge amount of iron, more than you need, which is obviously opposite of what you would be trying to do. So there, I cannot conceive of a situation where iron would become toxic in the environment.
So you mentioned the fish. So what is the response of a large algae bloom to zooplankton and then ultimately migratory fish in the ocean, non-migratory fish? I mean, one of the big ideas on how to make OIF more popular with people is to explain the cycle between iron, diatoms, Antarctic krill, and whales. So whale populations have been in decline for a long time due to industrial whaling and also a little bit of climate change. I think now that trend has reversed for most of the population, but it's still on a very small base, much smaller base relative to how many whales we've had 100 and 200 years ago. And also, whales have this magic whale poo where they basically do OIF naturally. So they poo in the ocean, have iron-rich poo, and then create their own algae bloom, and then create their own diatoms, create their own krill that they then, or copepod, and then they eat again. So I think just for the average human being, storing carbon in whale biomass is one of the most popular storage
Yeah,
systems for
yeah,
the climate problem. So we'd all have
yeah,
to have more whales. If I see a whale, it's just magical. They're
yeah.
very like popular beings for humans. So our core belief or core hope is that you need to promote OIF in a way that makes it popular so that it at least has a chance. So right now it
Mm-hmm.
seems to be one of the most controversial methods of carbon removal on the negative side, which in my point of view did not get enough funding, not enough resources in order to be evaluated whether it works. If you just compare it with something like direct air capture, which got a lot of funding, a lot of attention, and a lot of positivity, and didn't really, if you believe, like 2 billion was invested into a direct air capture, and right now it's still emitting more carbon than it removes. So possibly those past OIF experiments also emitted more
Mm-hmm.
carbon than it removed. Possibly, we don't know. It may have also removed more. I mean, obviously that's a question we want to find out. So in order to make OIF a little bit more popular, I do believe it might be valuable to talk about its potential co-benefits. And one of the most popular co-benefits would be to feed the whales. So basically, you're overfeeding the diatoms with iron. Diatoms
Yep.
grow in the Southern Ocean.
Yep.
Then Antarctic krill eats all those diatoms, and then suddenly the whales are not limited anymore by krill. You know, they have like a lot of krill to eat, and then they procreate faster, and
Yep. Yep.
we
Yep.
store all the excess carbon from the atmosphere in whales.
Yep. Well, I haven't done the calculation about how many whales it takes to get to a gigaton of carbon, but I don't think the ocean has enough room by the time we finish with this. So we're not going to win by storing carbon in whales, and this goes for fisheries too. And this is often to me a little confusing of why scientists say this, but you know, they talk about increasing the amount of carbon retention by increasing the number of fish. That's not going to do any mCDR, because that fish is either harvested and we consume it, and all of that carbon is remineralized, or as we say, and all of that carbon is released back to the atmosphere. Okay, so it may take a little bit of time, but it all goes back to the atmosphere. There is no recovery on any sort of a time scale. And even if we don't eat it, it usually is moving up trophic levels in other ways where it is recycled in surface waters. So there is no carbon. From a mCDR point of view, fisheries don't add to the carbon removal. Okay.
At least not 100 years. Just to be clear, like some fish may store it for a year or 5 years or 10 years
That's
or
right.
20 years.
That's right.
But not for 100 years.
But not for long-term storage, which is what mCDR is all about, is the long-term storage. That's why I say it's not, I don't know why people talk about it. At any rate, so how to make OIF more attractive? We've had this discussion. Not just with Exodus, but with other people as well. There are people that are suggesting OIF will increase fisheries productivity. And, you know, Victor is talking about whales. And that's, you know, it all makes intuitive sense. You make more, you know, phytoplankton are the grass of the sea. All right. So think about, you grow more grass, more things eat the grass, and it cascades up into the trophic level. So you should get more of everything, right? In theory, or not even theory, in intuition. Okay. But the ocean system is a lot more complex than that. And so scientifically, you cannot make a projection that you're going to increase fisheries, because we don't know whether it's going to get up there. Okay. You know, for example, and this is a very extreme example. All right. We have warming of the surface waters now. And we have more jellyfish blooms that are happening in the ocean. And more of those jellyfish are coming inshore, right? This is a big problem, because jellyfish are a dead end from carbon perspective, because not much eats jellyfish. Jellyfish will eat everything that's coming up from below. They'll take the carbon in, but nothing eats them. So they die, and all that carbon is regenerated, goes back to the atmosphere. It's a dead end. So you may increase productivity, but if it goes to jellyfish, you know, that doesn't help you from a fisheries point of view.
The whale's going to sink, though.
Well, but jellyfish, they don't sink fast enough. Okay, they're mostly water.
Yeah.
So they don't sink deep enough. They're not going to get by 500 meters is the point. Okay, they're going to be recycled in that upper area for the most part. Okay, and I'm not a jellyfish scientist, so if there are jellyfish scientists out there who say, no, you're wrong, I apologize. Okay. However, the point is that when we add more iron and we get more diatom growth, more phytoplankton growth, we can see a line that could go to more fisheries and maybe more whales. But that line has a lot of variability in it, where that could end up being very different. So we are not, as Exodus, we have decided we are not going to promote the idea of enhancing fisheries, because we don't feel that it's a scientifically sound perspective. We acknowledge that it might increase fisheries. There's a potential. And so that's what we basically say. And that, when people are really looking for a positive, saying there's a potential that it might do this is not necessarily very compelling to them.
Yeah. So my point of view on this topic is, you know, there's this like primary drawdown, sort of the direct drawdown, and then sort of the secondary. Primary hoping that it's sinking like 500 meters deep and locks away the carbon for 100-year plus. And then there's a secondary where there's some algae that maybe they only sink like 50 meters, 100 meters deep. Some of it gets eaten up by zooplankton. Some of it is just respiring and outgassing. So each carbon molecule definitely has a different path. So some may outgas after a year, some after 5, some after 15, 20, something like that. So it's like a big cocktail. They all have a different path until they may end up in the atmosphere. And the way I view the carbon industry is that there's two interesting metrics that for some reason have been chosen 100. So the idea is we want 100 years of permanence in order for like a carbon removal to be legit. And then we also want to pay $100. So for some reason, it's $1 per ton per year of storage. In my simple math, maybe there might be some benefit for a sort of carbon removal method that only costs $20, but it's only 20 years of permanence, and then you just repeat it every 20 years, and so you just stack it up, over 5 actions over 100 years, and then maybe even in 50, 60 years we have something like nuclear fusion, and then maybe that gets down the cost of energy a lot, and maybe technology with AI develops a bit faster. So we may not ultimately need those 100 years, and it may be enough to just like cool down the planet rather temporarily than permanently. So that's why I think it's probably even more complicated to measure, because the entire system is even more complex than sort of like this simplistic way of like algae cells sinking down to the bottom of the ocean. But there is potential in terms of it may unlock interest because people that, for example, do not believe in climate change or do not believe in what's called, like, I think Bill Gates today said, climate change hysteria is wrong, which is very different from what he said just 5 to 10 years ago. And I think this is sort of the current zeitgeist around climate change basically shifted a little bit. For like many years, it was marketed as like the biggest danger that we humans face, and I think that reversed a little bit. Right or wrong, I mean, it may be the biggest danger, it may not be, but people are a little bit like careful about it now, and I think the more we can package it as something beneficial anyways, the more funding and social license, etc., it may attract.
Right.
And then another thing that I've been, and maybe you have a good answer, maybe it's not true, but
actually, if I could, Daniel, before you go on, let me just deal with that initial question that you had, which is, what about if we take carbon out of the atmosphere for shorter times than 100 years? There may be some validity to that perspective, but okay, so think about this. The way that the system would work is that you have a surface of the ocean, you have a what we call a mixed layer, and we have what we call a permanent pycnocline, okay, that doesn't change throughout the season. So in the summertime, the pycnocline is up here, a new pycnocline is formed up here, so that you only have mixing in this top part. But in the wintertime, it mixes down to here. So we can tell if the carbon goes below this level, then it's not coming back each season. So it's away for at least a year. Okay. So now sort of zoom back out again, and you have all these different density layers going down. So the density increases as you get deeper and deeper. All right. And each of those layers basically mixes along the layer of density. Okay. And if you think about these layers like a dinner plate, it eventually comes up to the surface somewhere. And so that if the carbon sinks down to this layer, it will eventually move back up to the surface. Okay, and that may be in 10 years, maybe in 20 years, maybe in 50 years, depending upon how deep it is and where that intersects. How much time does it take to move along that line? Answer that question, to be able to calculate that, one needs to run the experiment to figure out how much carbon gets down to all of these levels. All right. So if you do the OIF, what is that what we call a remineralization? Remineralization just means you take an organic particle and you turn it back into its mineral components, the nutrients, the CO2. So it depends on that remineralization length scale, we say. So how much of it happens here, how much here, how much here, how much here, how much here. And that, by knowing that, by measuring that with OIF, which is a non-natural bloom, by measuring that, you will have an idea about how much of the carbon is going to be in this layer, how much in this layer, how much in this layer. So you'll be able to say how much carbon is going to be away for what lengths of time. At that point, then you have data in which to decide, does it make sense to take carbon away for 50 years? Based on the amount of carbon that's going to be taken away for at least 50 years, that in this layer and deeper. But right now, we just can wave our hands with estimating it.
Okay. One thing I always wonder in that context of fish is that if I'm a big fishing company and I fish in the Southern Ocean, okay, and I'm taking out fish, technically I'm taking out iron, right? So I'm technically taking out iron from the system. So one of the possible distribution methods for iron in the future, let's say OIF is validated and we've measured it and we like it, is just to introduce laws that if I'm taking out iron as a fishing company, I kind of need to put it back in by like adding a little bit of iron on my journey. Does this make sense in some sense to you?
Not really, because the amount of iron that we're taking out with fish, we take out a lot of fish, and it does equate to a big amount of iron when we look at it. But when you compare it to the amount of iron that's naturally going into the system, it's infinitesimal, and so it wouldn't make much of a difference. Half of the iron, say in the subarctic Pacific, half of the iron is coming from the atmosphere. It's coming from dust. The Asian deserts, Gobi Desert and others, the dust comes across and settles into the ocean, and that's where half of the iron's coming from. The other half is coming from water that's from below, the mixing upwards of iron from the deep ocean. And the deep ocean iron comes from the dissolution of the dust going down. So that's where most of the iron is coming from. And the fish that you take out has such a small proportion of in comparison to that it wouldn't make any difference.
Even if you'd consider it historically, even if you'd consider all the iron that got out taken like for the last like, I don't know.
Yep. Even if you consider it historically, because you have to consider historically the amount of iron that's coming in by dust, right? So you go year by year by year, and you say, okay, well, this year we would bring in, I can't even get small enough fraction of the iron that's coming in the fish versus coming in the atmosphere. Well, each year it's the same. So it's still, you wouldn't be making any difference. And then basically you're doing an ocean iron fertilization. So is that
would be
is that a good idea?
Evidence.
We're back to square one.
Post evidence. Like let's say in 20, 30 years we believe it's good, but we want to figure out how to get in iron cheaper into the ocean. And there's just so many fishing boats on the ocean. Just figure, okay, why don't you put in the iron that you've taken out? Just like
and that may be ultimately in the long term. That could be a way of going at things, but one of the issues is if the carbon. So one of the enticements of this would be that the fishing boats could get carbon credits for doing this. But in order to get carbon credits for doing this, you have to be able to verify the amount of carbon that's being removed from the atmosphere. And that's a key part of these planned experiments, is figuring out what do we measure, what can because we can't measure everything. If we go to 1,000 kilometer by 1,000 kilometer scales in implementation, we can't measure everything everywhere. So what subset of measurements can we make that would give us an accurate estimate of the amount of carbon that's been removed for X amount of time? And once we have a better idea of how to do that in a logistically feasible way, then one could look and say, well, with the fishing boats distributing iron, can we actually verify how much carbon is being removed by the amount of iron that they're adding? And at this stage, there's no way we can. But perhaps in the future, we would be able to use a subset of measurements to be able to get at that.
Yeah, same for like ocean freight logistics and anyone sort of who's emitting on the ocean by like fuel. Maybe in the future there's like a carbon-free fuel or like some better batteries. more efficient
Yep.
hiking
Solar.
method.
Solar's going right
So,
now. They're starting with solar, and ultimately that's the way things are going to end up going. And the shipping industry has a huge incentive to try and make this work because a major portion of their costs are fuel. So if they can, switch over to battery solar operated systems, then they're far better off.
So maybe if you were to estimate, if you were a betting man, okay, and you'd have to sort of like draw like a prediction market on, will there be an ocean iron fertilization experiment that is giving us new parameter, or like feeding the models and informing us on the feasibility, effectiveness of OIF. What would be your estimate? Like, what's the percentage it's going to happen in 26, 27, by
No, it's a good
2030?
question. I'm not very good at betting, so I don't gamble. Okay. I would say that the chances are better than half that there will be an experiment done sometime by the end of the decade. Okay, I'll say that. The question really is, can we come up with enough funding in order to mount a meaningful experiment? There's no point going out and doing this on too small a scale. We're wasting our time. We're wasting money. So it needs to be done on a scale that's on the order of 30 by 30 kilometers or 50 by 50 kilometers. And so if we can't get to that stage, there's no point in doing it. I will say that we are moving. One of the big issues is how to get the iron out there. Technology, and this is a big difference too when you compare it to the previous experiments. Technology has changed tremendously since those mesoscale enrichment experiments. We now have profiling floats. We have gliders. We have remotely operated AUVs, so things that perhaps can be distributing the iron, for example. We don't have to use a ship anymore. We can use a fleet of AUVs to do this. We have drones that can monitor the development of the bloom, at least in the surface-most waters. So there's lots of technology that is improving right now that will decrease the cost of actually implementing this. So as we go over the next, say, 2 to 3 years, we're going to see the costs necessary in order to run the experiment going down. At the same time, hopefully we're building the momentum to try and run the experiment going up. And it's when those two intersect, I'm not going to predict.
Fantastic. So yeah, from my side, I think I covered most of the topics you want to discuss. Is there things that you feel are important that we haven't really?
Not really. I guess what I'd like to do is emphasize that. Emphasize a couple of points. The first is that none of us who are working on this would like to be working on it. Okay, we're working on it because, frankly, we're very worried in the long term. I've got kids. Most of the people I'm working with have kids, and we're thinking about what kind of a world are they going to be dealing with moving forward. Our generation has really screwed things up badly by not cutting CO2 emissions. And I think the least we can do is explore the possibility of removing where those possibilities are. They need to be looked at. And we're not, as scientists, we're not doing our job unless we do look at it. And to make arguments that we shouldn't be looking at it, period, without knowing what the outcomes of those experiments might be, is, in my view, irresponsible. Okay, as scientifically irresponsible. That's not to say that ocean iron fertilization should be implemented. I have big reservations about it. But I think we need to get answers about just how bad an idea is it. Okay, and then put that into comparison of what the projections are if we're not successful. Because right now, the only method out there that could approach the 10 gigatons, or it won't be 10, but approach multi-gigatons a year of carbon removal is iron fertilization. There is no other method that would approach that, no other single method that would approach it. Okay, so that's the first thing I would like to emphasize. The second thing is that all of the previous mesoscale iron enrichment experiments tell us about how the ecological system is going to respond, because we've got basically uniform response to the ecology. It doesn't tell us about what magnitude of carbon exports we're going to get and what the shape of that export looks like, how deep, how much, so that we can't use those experiments to answer the questions that we have now. We need to run experiments that are designed to answer the questions we have now. Okay, so that's the second thing I'd like to implement or point out. The third thing that we haven't talked about is to recognize that the impacts from any implementation scale iron fertilization are going to be both the ecological, it's going to be economic. There will be winners and there'll be losers. There will be cultural components, because there may be systems may change in a way that have direct impacts on the cultures of individuals, whether it be fishing cultures, whether it be indigenous cultures. There's going to be broader societal impacts that all need to be considered. Okay. Now, none of those impacts are going to be significant with experiments. All of them might become significant at implementation. And so we need those experiments run so that we get a better feeling for what those changes will be and what that might mean. But none of those are reasons to not experiment, to look to see how well might this work and how bad an idea is it.
Fantastic. I really hope you find the money. I really hope this is going forward. Personally, I believe that there might be a world in which any funding source is okay. I personally disagree with the fear that taking money from industry like Microsoft or from venture capital itself will make the results non-credible. I feel that all of the efforts of carbon removal have taken industry money, and that necessarily didn't make their data any non-credible. I mean, like Climeworks get billions and they still couldn't remove more carbon than they've been emitting in the process of trying to remove the carbon. So, and it came out, and people were like, okay, so maybe direct air capture doesn't work yet, and it was sort of okay. So I feel that in general, the industry needs to be a little bit like less self-critical. It's like the moral bar, the moral standards was way too high. My biggest fear, to be honest, is that it's not going to happen at all, because there's not enough flexibility to just say, okay, we'll like just try to find any funding source possible. We try to maybe start with a smaller experiment just to see if people are as enraged as they have been in the past, and just trying to work in a step-by-step function. So my current view, or my fear is, like my intuitive fear is that the bar has been raised so high that it's everyone's speaking about like this one big experiment that will cost a lot of money, that will change everything. And that's not necessarily my if I study history, my theory of change isn't necessarily that this is always how things work. Sometimes it's sort of like you need to start small and you need to repeat and you just need to iterate, and actually, like, let's say 1 million helps to attract 5 million, and 5 million helps to attract 20 million, and that ultimately gets you to the 50. So, obviously, any progress is good progress. Talking to anyone about the subject is positive and inspiring. I think young people, young scientists, young funders, young politicians to look into OIF as an option is a positive. So I thank you so much for your time and your deep wisdom and your effort in the space.
Well, thank you very much. It's a pleasure being here, and I've enjoyed this very much.